Good morning students. Today we are going to perform the simple staining method. For simple staining we require, as you know what is the staining method. So we require a culture and methylene blue, that is the basic stain, and I take the second one is, this is the methylene blue solution we require, and this is the eosin blue, that is it is a acidic stain. This is the basic stain we require. And today we are going to perform, I wipe this area working platform with the ethanol as it is a disinfectant. Okay, and I take the culture of bacteria. I take the slide. This slide is there. So this slide is grease free, so we require this slide. And now we will perform the practical. Okay, so see here, it is a nichrome wire loop and it is red hot. That is, it is a sterilization process. Red hot is there. See here, it becomes red hot. Why we require this nichrome wire loop? Because its melting point and its cooling point is very less. So we require this. And this is the slide, which is grease free slide I take. Okay, so I prepare the suspension first. I take a drop of the water, okay, on the slide. Okay, then again I red hot it, sterilize this loop and take out this colony from this. I take out one colony from this new plate. Okay, and now I will prepare the smear. So I am preparing the smear here. The smear which you have to prepare, it is very thin. Okay, and if it is very thick, then we cannot observe the morphology. So we require this thin smear here. So I am spreading this smear. See here, the smear was, before keep this, it again sterilized. See here, the smear was ready on the slide. Okay, see the smear. Then wait for drying. Once it become dry, you have to heat fix it. Why we are going to heat fix? Because the morphological characters are to be settled down. Okay, and the staining become proper here. So we prepare this smear. Okay, and once the smear is formed, then just heat fix it like this. 1 2 3. Okay, so now it is ready. See here, the smear is there. Are you observing this smear on the slide, white color smear? Okay, so you have to prepare this smear and then you observe under oil immersion objective in the microscope. So we require the cedarwood oil. Okay, so I will prepare the next slide. One is for monochrome staining and another is for negative staining. Okay, so we are going to perform the simple staining method. In that the monochrome staining is there and as well as negative staining is there. So this is the slide I take. The smear was same for each and every staining method. Okay, you have to wait for its red hotness. See here, it becomes red hot, and then wait for cool down. It will cool down very fast. Then you take out the colony from this. Just take out the colonies. Actually this colony become very dry, so here I am preparing the smear. See here, this slide is not grease free, so the smear was not preparing very well, so I am doing again. I will take again one colony and mix it with this. Now it's the smear was formed. Once the smear was formed, then again I will sterilize this loop. Okay, keep aside the loop. The smear was formed. You can see the smear here on the slide. Wait for drying. Okay, the same procedure is for monochrome staining, same procedure, smear making procedure for monochrome staining, from negative staining, for gram staining. You have to prepare this within this burner. I am working within this burner, and you have to prepare this smear, very thin smear. See here, the smear was observed on the slide. Okay, once it become dry, then heat fix it. Okay, so these two slides are ready for staining method. So the staining method, it is a method of microbiological method which can observe. By this way we can observe the arrangement of the bacteria, the morphological characters of the bacteria, how they are arranged, and either they are diplococci or either they are oval shape. All these things we are getting understood by the staining method. We are going to perform the simple staining. Simple staining are of two types, that is the monochrome staining and negative staining. So in monochrome staining we are taking the basic stain. What is the basic stain? Basic stain which carry the positive charge on it. So this cell, we prepare the smear, so on that there is a cell present. The cell carries negative charge. So negative positive get attracts and it become color. The cell become color. And in negative staining, here we are using the acidic dyes or acidic stain. The acidic stains are carries negative charge, negative get repels, and here the background become color and cell remain colorless. Okay, so this is the methylene blue. The methylene blue chloride is the name of this stain. It dissolves methylene blue positive, chloride negative. So it is basic stain. We are going to add this on the smear. Okay. You just add 1 to 2 drops of this stain, then wait for 2 minutes. Okay. The second one is eosine blue. See here, eosine. This is acidic stain. Just add 1 to 2 drops. Don't add excess of the stain. Then your figure or your morphology of that particular microorganism is not observed. Okay, so wait for 1 to 2 minutes. See here, this is for acidic, this is for basic stain, that is basic stain is added. It is for monochrome staining and it is for negative staining. See here, this blue color is there, it is of methylene blue, and it is for monochrome staining, and it is the eosine is added on this slide, so it is for negative staining. Now we will wait for 2 minutes. After 2 minutes I will discard it into the basin, and then I will wash it with tap water. Okay, so The 2 minutes are over. I will discard it. Okay, discard it into the basin, and I will add the water drop by drop. So drop by drop I will add. If you added the water very forcefully, then it will be the smear will be discard from the slide. So gently wash the slide. So I will again wash it properly. Okay, the washing is done. Now I will wait for its drying. Okay, I will keep aside this. Then I will discard the stain on this also, the negative staining, and wash it by drop. So the washing is there, so you have to wash like this. Then now we are waiting for the drying. Okay, so here is the microscope. So how to adjust the microscope? See here on this, it is given 100x is there, 45x is there, and 10x is there. So we have to adjust the microscope on 10x first, then adjust the highest intensity of light. Okay, you have to adjust the light at high level. Okay, so I adjust it at high level. Then take your slide. So this is the slide. Okay, and then take the cedarwood oil. Okay, so I take the cedarwood oil here and add 1 drop of cedarwood oil. It is sufficient. Okay, and then observe under microscope. So how? See here, I keep this here. Okay, and then the oil immersion objective is dipped here using this. See here, it is dipped now. See here, this is dipped within the oil. Okay, now I will observe